you very much for coming to this week's British Heart Foundation Centre of Excellence talk. Today, it's a pleasure to introduce Samantha Salvage, who works in Tony Jackson's laboratory in the Biochemistry department. But Samantha has, right through her career, starting from working in Surrey and working in China and Australia, managed to maintain a continuous theme relating to molecules that mediate conduction of the action potential and its velocity, as well as its potential, therefore, to generate a rhythmic substrate. Samantha will be talking mostly about one of these molecules, which is a sodium channel, which and what regulates it, and how it may lead to electrophysiological mischief. Although I know she will finish with taking the story a bit further and telling us where this particular episode might go. So with no further ado, let me let Samantha take the stage and take us through her very connected story.
Thank you, Chris, and thank you for the opportunity to speak here today. As Chris mentioned, I'm going to be talking about some of the work that I've been doing in Tony Jackson's lab, and that is on sodium channel complexes and cardiac arrhythmia. I'll start by giving a brief overview of NaV1.5 function in relation to cardiac excitability and conduction, and then look at how the beta-3 subunit affects NaV1.5 function, and hone in on some of the structural aspects of the beta-3 subunit that mediate these functional effects, in particular focusing on some key mutations, and in particular a novel mutation associated with Brugada syndrome. And then from there, we'll look at how the beta-3 subunit is involved in higher order clustering and some non-canonical adhesion functions, which have implications for cardiac conduction. And that's where gap junctions will come into. So the cardiac action potential is the electrical signal which initiates the events leading to contraction. It's controlled by the sequential opening and closing of a variety of different voltage-gated channels. And in particular, this rapid depolarization or upstroke is controlled by the sodium current through NaV1.5 channels. NaV1.5 is encoded for by the SCN5A gene, and to date, in more than 100 mutations have been identified in NaV1.5. And in particular, those inherited mutations which are associated with loss of function are associated with Brugada syndrome. And those that confer gain of function are typically associated with long QT3. But there's also aberrant NaV1.5 function in certain acquired cardiac disorders, including myocardial infarction and cardiac failure. But not only is it important that this signal is successfully generated, it has to be propagated across the entire myocardium in an orderly fashion to enable the syncytial contraction of the atria followed by the ventricles. And this proceeds in a manner akin to cable theory, whereby you get the passive propagation of local circuits down the fiber. Specifically within cardiac myocytes, this is facilitated by gap junction channels, which connect neighboring cardiac myocytes, providing a low resistance pathway for the depolarizing wavefront to traverse. So looking specifically at the NaV1.5 structure and how it relates to function, the primary structure is shown here, and it's one long single polypeptide around 250 kilodaltons. And within it, you can see it has this four-fold pseudosymmetry, so creating domains 1, 2, 3, and 4, which is shown here in a 3D structure of the cryo-EM structure. Within each domain, there are these transmembrane S helices, 1 to 6. The first 4 comprise the voltage sensing region, and then the neighboring 2, 5 and 6, and their re-entrant pore loops form the pore. So these pore domains sit here near the central region, and the voltage sensors of the same domain are concatenated out to the periphery. In particular, within the voltage sensing region, the S4 segment contains a run of positively charged amino acids, such that when the membrane depolarizes from an arriving electrical signal, the inside becomes less negative, and this segment moves outward through the membrane, pulling on this cord and therefore causing a conformational change in the S5 and 6 helices. This causes rotation around the center such that the channel opens and sodium ions enter into the cell, causing that rapid upstroke. And this occurs within all the domains. Subsequent to opening, the channel rapidly enters an inactive state, and this is largely controlled by movements within domain 4, the S4 segment of domain 4. This allows changes within the IFM motif, which bind to an allosteric site and prevent further entry of sodium. After this, the channels will recover and return to a closed state where they are ready to be excited again when the next signal arrives. Now, while the sodium channel, NaV 1.5, is fully functional by itself, its activity is fine-tuned by interacting beta subunits, of which there are 4 types, beta 1 to beta 4. But all have this much more simplified structure compared to NaV 1.5. They have a large extracellular domain, which is in sequence, it's similar to an immunoglobulin-like domain, so a lot of anti-parallel beta sheets, connected by an extended flexible neck to a single transmembrane alpha helices and an internal C-terminal region, which is largely disordered. We've been particularly interested in the beta 3 subunit for a number of reasons, but one key one is that its knockout in a murine model produces this severe arrhythmic phenotype. And indeed, mutations throughout the beta 3 subunit are associated with a variety of diseases, including atrial fibrillation, Brugada syndrome, and sudden death. So we decided to look at the effect of this beta 3 subunit on NaV 1.5 function in HEC293 cells. So we have NaV 1.5 stably expressed, and we transfected an empty vectored GFP in order to look at the control condition, compared to a beta 3 with a GFP tag. So this is patch clamp electrophysiology, and effectively we are clamping the cell at -120 millivolts as a resting potential, and then introducing successive depolarizing pulses, which increase in 5 millivolt increments. As these increase, you start to cause the channel to open and currents to appear. As this becomes more depolarizing, the currents increase up to their maximum and then decay away. When you compare these two visually, you can see that it doesn't look like there's much difference. The maximum current is largely the same. And looking at the accumulated data, when each current is plotted against the voltage which elicited it, so the IV curve, we can see peak current is indeed the same, so beta 3 does not affect this in the HEC293 system. And there are no voltage shifts. So largely we can say that the beta 3 subunit is not affecting the activation properties. So we looked at the next state, and that is steady state inactivation. To test this, you introduce a conditioning or a pre-pulse, which varies through different depolarizing steps, followed by a test pulse at -40 millivolts, which is well within the activating range for the NaV 1.5 channel. So these first pre-pulse steps are very hyperpolarized, around -140 millivolts, such that this test pulse produces the maximum current. This persists for a while, but as this pre-pulse becomes more depolarizing, channels are pushed into an inactive state and the amount of current decays away. Now here you can see that the presence of beta 3 is causing a rightward shift, or a depolarizing shift of around 5 to 10 millivolts. This has the effect of increasing channel availability at any given voltage step compared to the 1.5. Or in contrast, you could look at it that the 1.5 without beta 3 has less channels available. This is akin to a loss of channel function and could be something similar seen in Brugada syndrome. So then we looked at the final state, recovery from inactivation. This is tested by delivering a -40 millivolt pulse, followed by a second -40 millivolt pulse at increasing time intervals. So this first pulse produces the maximum current, and the second pulse will produce a smaller current until enough time has passed for it to reach back to the maximum. So you plot the second pulse as a fraction of the first over the time taken. And these two graphs are just shown on different scales to emphasize the effect of beta 3. What you can see is that the presence of beta 3 accelerates this recovery from inactivation process. And one final thing we looked at, moving on from steady state properties, we looked at the kinetics of the activation-inactivation process. So we did some Hodgkin-Huxley modeling, and this uses an m-cubed function, which represents the 3 activation gates, and the h function, which is the single inactivation gate, and derive time constants for these parameters. What you can see is the presence or absence of beta 3. These curves are largely similar, so the time constants of these processes does not change in the presence of beta 3. This means that the functional cooperativity of Nav1.5, so the propensity of another channel to open as one is activating, is largely unchanged. Overall, this means that the beta 3 is fine-tuning the action of Nav1.5, but largely within the properties of inactivation and recovery from inactivation. So we were interested to know within the beta 3 subunit which components of the protein are conveying these functional effects. And so if I just remind you, there's a large extracellular-like immunoglobulin domain, which in itself gives a lot of implications for cell adhesion functions and potentially some immune functions, and a large area for contact with the extracellular face of the Nav1.5 protein itself. And then there's the transmembrane alpha helix, which could be binding at any site around the Nav1.5. So these two areas were of interest to us. We did a sequence alignment, and the transmembrane region is highlighted here in gold, and again in this visual depiction here. And within this, what we found is there is a large level of conservation across species. This is highlighted by the magenta sequences and the asterisks down here. Now, the transmembrane region is a hydrophobic environment, and it makes sense that most residues will be largely hydrophobic in nature. What was quite surprising is that there's this highly conserved glutamic acid, and it's present in all these different species. There's an equivalent residue also present in beta 1. And it's energetically unfavorable for a residue like this to exist in the lipid bilayer. When residues like this occur, such as those positively charged amino acids in the S4 segment of Nav1.5, if they're conserved like this, they often have some important function. And indeed, in beta 1, it has been shown that this residue is involved in some of the gating effects on other sodium channels. So we decided to investigate this further. We took the wild type beta 3 and used an EGFP tag just to make them ease for detecting the protein. And to determine whether the glutamic acid is involved in any electrostatic interactions, we reversed the charge by switching it to a lysine to disrupt any of those potential interactions. And in addition, given the likely importance of the extracellular domain, we created a construct lacking this region, and then a combination of the lack of the extracellular region plus that point mutation in the transmembrane domain to look at multiple points of interaction with the alpha subunit. So the first thing we did was just to check, did any of these mutations disrupt the alpha-beta interaction? We carried out an immunoprecipitation. The Nav1.5 subunit is tagged with an HA, and we used an antibody to this to pull it down. So this is the bound fraction, and this is the unbound or the flow-through. And you can see 1.5 was pulled down in all samples, and with that came the full-length beta 3, the E to K mutation, and then those beta 3 subunits lacking the extracellular domain itself alone or together with the E to K. So this highlights that there are multiple points of interaction throughout the beta subunit that maintain that interaction with Nav1.5. What's quite striking in this plot is the density of the signal produced by the delta ECD. And that's likely because such a large immunoglobulin-like domain on the extracellular face is probably constricting how many beta subunits can bind to the alpha. But without that large region, then your transmembrane domain is probably free to bind to many different sites, and hence you pull down more of this subunit. So naturally, now that we know that they are still interacting, we decided to look at function using patch clamp electrophysiology. And with all of these mutations, looking specifically at activation, you can see that peak current is unchanged and there are no voltage shift changes in activation. This is pretty much unsurprising, given that previously I've shown you that the beta-3 has no effect on these properties. So what about inactivation, where beta-3 increases channel availability? Well, when you look at that point mutation, the glutamic acid to lysine in the transmembrane domain, there is no change to this depolarizing shift induced by beta-3. So this still confers an increased channel availability. In contrast, however, when you lose the extracellular region, this shift is completely abrogated, which implies that it's the extracellular region of the beta-3 subunit that is probably sitting somewhere near the domain 4 voltage sensor to mediate these shifts. And then we also looked at recovery from inactivation. So as before, beta-3 accelerates this process. Now we find that the E to K mutation in the transmembrane domain and the loss of the extracellular domain both confer a loss of acceleration, which implies that perhaps the transmembrane region is binding somewhere in proximity of domain 3, close to that S3-4 linker that's controlling recovery from inactivation. So we looked at the voltage sensor movements in a little bit more detail, and this was some work carried out by our collaborators at Washington, Professor John Silver and Dr. Wendy Zhu. In order to selectively look at specific movements of voltage sensors within given domains, they used a process called voltage clamp flowmetry. And this was carried out in cut open oocytes. Essentially, you can track the movement of the S4 segment by introducing a cysteine residue in proximity of that S4 voltage sensor. So that was just in the S3-4 linker above this, and labeled it with MTS TAMRA. When the voltage sensor is activated and moves outward through the membrane, you get a change in fluorescence, which is tracked and then normalized. So this was first carried out for the domain 3 voltage sensor. And here we see that the presence of beta-3 causes a rightward or depolarizing shift. So the activation of the voltage sensor occurs later. This shift is lost with the presence of the E to K mutation. Similarly, the delta ECD also abrogated this effect, but actually made things more hyperpolarized. So then they looked also at domain 4, given its importance for inactivation. And here we find that the presence of beta-3, irrespective of the E to K mutation, causes the voltage sensor to activate later, but largely it catches up and merges with the lack of the beta-3. In contrast, however, losing the extracellular domain causes a rightward or depolarizing shift of this voltage sensor movement. And it's probably this which conveys the effects on steady state inactivation. With the E to K mutation, it sits somewhere in between the two. So overall, from the effects that we've seen on recovery from inactivation and steady state inactivation, as well as these voltage sensor movements, we can see that the transmembrane region is important in mediating effects in the kinetics of recovery from inactivation through domain 3, and that the extracellular IG domain is important for both processes, but additionally this steady state inactivation shift, and so is mediating effects on both domain 4 and domain 3. So given the importance of this extracellular domain in mediating many of the NaV1.5 channel functions, it was quite interesting to us when we were presented with a patient who was experiencing palpitations and dizziness and presented with an intermittent type 1 Brugada pattern ECG. So this is coved ST segment elevation and an inverted T wave. This was provided to us by Dr. Greg Meller at Papworth. And what was particularly interesting is that when next generation sequencing was carried out on a host of cardiac ion channels, typically associated with Brugada syndrome, in the key channels, nothing was found, including SCN5A for NaV1.5. But interestingly, the only mutation present was a novel in-frame 3 base pair deletion in SCN3B, the gene encoding beta 3. And this worked out to correlate to the loss or the in-frame loss of a single threonine residue at position 138. That's highlighted here, and it exists slap bang in the middle of the beta antiparallel beta sheets. So you can imagine if you pull that residue out, all subsequent residues are going to be pulled out of alignment and disrupt the normal hydrogen bonding partners and any other potential ionic interactions or others. So we decided to engineer this mutation into our beta 3 construct. So we have the wild type beta 3, the beta 3 lacking that threonine, and then we also carried out a co-transfection equal mixture of the two in order to try to recapitulate the heterozygous nature of the individual. And while these aren't high resolution, what you can see is they largely express well and seem to be distributed to different membrane compartments, in contrast to just the extracellular EGFP, which is diffusely spread across the cytoplasm. Then, of course, the first thing we decided to look at was the function. Carrying out patch clamp electrophysiology, what you can see quite noticeably here is that the currents with the loss of that T138 are noticeably smaller than the other conditions. And when you look specifically at peak current, you can see that the loss of the T138 results in a reduction of current by about 50% to 2/3, which could account for Brugada syndrome. However, when you co-transfect the wild type beta 3 with the loss of that threonine 138, suddenly the current looks more like just the beta 3 on its own, which wouldn't really account for Brugada syndrome. So we also looked at inactivation. And as always, beta 3 has caused its depolarizing shift, so an increase in channel availability. And then losing that threonine 138 causes the curve to converge back on the NaV1.5 itself, as if the beta 3 isn't there. And this is true also for the mixed expression of the wild type beta 3 with the T138 deletion. So that could go some way to explaining a loss of channel availability in this individual. But given that the loss of the T138 seemed to have such a marked effect on peak current, we wondered whether this might be in some way reducing the forward trafficking of NaV1.5 to the membrane. So we looked at the expression of the beta 3 with and without the mutation and NaV1.5 in the cytoplasmic and membrane compartments. And here it looks like, although they're all massively overexpressed as expected for a transient transfection, the wild type beta 3 seems to be more abundant than the T138. However, this didn't correlate to a change in total NaV1.5 expression. They're largely similar, or if anything, the NaV1.5 was actually more abundant in the deletion of the T138 sample. So we just first of all double checked that each of these mutations was able to still interact with NaV1.5. So we did a pull down of NaV1.5 and then found that the full length wild type beta 3, the loss of the T138 and the co-mixture all came down largely to a similar extent, despite their differences in relative overexpression. So that suggests that interaction is saturated. Now, the previous data showed that the membrane expression of NaV1.5 was largely similar or perhaps higher in the T138. But that's all membranes and could incorporate things such as the Golgi or the ER, where perhaps some channels are trapped within the secretory pathway. So we carried out a cell surface biotinylation experiment to selectively label only those proteins at the plasma membrane. So the cells were incubated with a non-permeant biotin at 4 degrees to try to reduce any endocytosis. And then the cells were quenched, lysed, and a streptavidin pull down carried out to pull down biotin specifically. So the biotinylated fraction represents that at the cell surface, and this non-biotinylated fraction is anything that was internal. And here you can see that the beta-3 full length with the loss of the T138 or the co-mixture all get to the surface absolutely fine, as does NaV1.5. So this tells us that the loss of the threonine at position 138 in the extracellular region is not perturbing the trafficking of NaV1.5 to the surface, and therefore not accounting for any reduction in peak current that could contribute to the Brugada phenotype. So one final thing we looked at for this mutation was to try to determine if losing that threonine residue causes any gross structural disturbances to the immunoglobulin-like domain. So for this, we purified just the extracellular region for the wild type and for the loss of the threonine residue, and subjected the samples to circular dichroism spectroscopy. This technique allows you to determine the secondary structure that is present in particular proteins, and different structures give characteristic spectra. So this here for beta-3 wild type and for the T138 deletion are largely superimposable within error, and is characteristic for a structure that is dominated by beta sheets, which typically peaks in this region of 210 to 220 nanometers. If we had something like random coils, we'd get a negative deflection and peak around 195 nanometers, and alpha helices cause a double peak around 205 nanometers and 225 nanometers. So that's characteristic for an immunoglobulin-like structure, and largely that is not affected by the delta T138. So something we wondered was whether there would be any temperature disturbances to this, because what was quite interesting is that the individual in question had actually previously presented with a Brugada type ECG when they'd been in the febrile state. So we wondered whether increasing temperature might cause some destabilization. So to do that, we carried out a thermal melt profile where temperature was increased by 2 degrees at a time, and the wavelength was monitored at 217 nanometers in the middle of what is typical for a beta sheet. With increasing temperature, we found that there was actually no change to the structure. There was no denaturation, no unfolding. It was incredibly stable, irrespective of the presence or absence of that T138. So this suggests to us that the loss of that threonine residue is probably causing more local disturbances to protein-protein interactions rather than any gross unfolding or destabilization of the protein. Now, this is quite interesting because this single loss of the threonine was able to abrogate steady state inactivation in a manner similar to losing the whole extracellular domain. So it suggests that this region of the immunoglobulin domain is quite important for protein-protein interactions. So that could be with both the NaV1.5 itself, as well as other beta subunits or interacting proteins. And this brings me back to the idea that the beta-3 Ig domain could be involved in cell adhesion functions and potentially some immune functions. And what's quite interesting is that in the group previously, the beta-3 Ig domain was found to form higher order oligomers. Particularly, it could form trimers. So what we didn't know is this true of the whole beta-3 protein? And what implication does this have for NaV1.5? So we first checked, could the beta subunit form binos by using a beta-3 tagged with GFP and a separate beta-3 tagged with MYC. And indeed, when we pulled down the GFP, the beta-3 GFP and the beta-3 MYC came down. We then carried out some chemical cross-linking using the non-permeant BS3. And in the absence, we see this single monomeric band followed by a smear, which is indicative of the heavy glycosylation that occurs with beta-3. And this is the same for beta-3 MYC. The differences in migration are simply down to the different sizes of the tag. Once the crosslinker is added, and the sample is cross-linked via its lysine residues, we start to see bands at around 120 and 180 kilodaltons, which are indicative of dimeric and trimeric forms. So indeed, the beta-3 subunit can form these multimers. But what does that mean for NaV 1.5? So we carried out a proximity ligation assay. This technique works that you can determine whether two proteins are in close proximity, so that's less than 40 nanometers of each other, by using different species of antibodies, so that when you come in with the oligonucleotide probes specific for those antibodies, if they're closer than 40 nanometers, you can add a substrate that will allow for roll-in circle replication amplification in a manner akin to PCR. And given that the probes are labeled with a fluorophore, you get these intense areas of dots that are detected by the fluorescence. So we did this by using two different NaV 1.5s in order to specifically look at their proximity. So one was labeled with HA and one with GFP. In the absence of beta-3, you can see the presence of the GFP signal, and in the corresponding PLA channel, there's very little that you can see by eye. However, when you add beta-3, suddenly the signal goes up dramatically. And when this was normalized for multiple regions of interest, we find indeed the number of dots in the presence of the beta is far greater than in the absence, and the intensity of the signal is also increased. So this suggests that the beta-3 subunit is somehow increasing the ability of the NaV 1.5 to form these higher order clusters at the cell surface, and that is shown by these transitions here. So we went a little bit deeper with this and decided to do some single molecule localization to try to determine exactly how many sodium channels are in clusters and how they organize throughout the membrane. For this, we use stochastic optical reconstruction microscopy, or STORM. This takes use of special properties of certain fluorophores. So here we used Alexa 647, which has the capacity to photo switch or blink. It goes from an on state to an off state where it remains. And so effectively the fluorophore blinks only once during the experiment. And it allows you to get this time-resolved sequence of blinks, and therefore the precise location of where the protein is. So we did this for NaV 1.5 on its own and in the presence of beta-3. And what we found, in contrast to the proximity ligation assay experiments, is actually that the density of sodium channels at the membrane and the observations in cluster was largely the same. So this tells us that NaV 1.5 is able to form these higher order clusters, even without beta-3. So our collaborators at Harwell did some further analysis and looked at the cluster radii and the nearest neighbor density. And here it's where things got interesting. In the absence of beta-3, we see a peak around 6 nanometers. But as soon as beta-3 is present, the peak shifts by about 6 nanometers. So this is true also for the nearest neighbor density. So this tells us that NaV 1.5 has the capacity to self-oligomerize. However, the beta-3 causes subtle rearrangements to the geometry of these clusters. So that made us wonder what the implications of this are for cell adhesion, and in particular, cardiac conduction. So just to remind you, electrotonic conduction, which is the mainstay of cardiac conduction, works by the passage of ionic currents through low resistance pathways connecting adjacent myocytes, so that the depolarizing wavefront can be propagated from cell to cell in an orderly manner. But what has become more apparent recently is the idea of this electrical field transmission, or ephaptic conduction. Now, this process works by the idea that there is a narrow cleft between adjacent cells, and within this there is the expression of the NaV 1.5 proteins, such that when the first cell, cell 1, is excited and depolarized, these sodium channels open. Now, when this one here opens, it will withdraw sodium ions from this narrow cleft. And because this cleft is so narrow, this will actually alter the transmembrane potential across the quiescent cell too. So if you imagine that sodium is withdrawn from here, this will become less positive, so the inside becomes less negative, and this will allow the S4 positively charged segment to move out through the membrane and open the channel, thereby taking sodium into this cell and causing depolarization, which means that the depolarizing wavefront has been conveyed to cell 2 without ever having actually passed any ionic current directly from cell 1 to cell 2. Now, there are some very crucial physical constraints on this mode of conduction, and those are that you have to have a significant number of Nav 1.5 channels in this particular region. And it has been known for a number of years that the density of sodium channels at the intercalated discs far exceeds what is necessary for electrotonic conduction. The other important parameter is the distance between neighbouring cells in order for this electrical field to build up. And it's been modelled that this should be in the region of approximately 20 nanometers. So quite interestingly, when a couple of years ago, a group looked at the role of the beta 1 subunit and found that Nav 1.5 bound to beta 1 was able to form these trans cell adhesions, which actually facilitate this approximately 20 nanometer gap, was really quite fascinating. And when you take this together with the data I've shown you today, that beta 3 performs these roles in cis adhesion. So you can imagine these complexes sat in one cell membrane being facilitated by beta 3 interactions. You can see how both trans cell adhesion between beta 1 and cis adhesion between beta 3 may help to stabilize complexes both within one cell membrane and those at the opposing one to enable the formation of this ephaptic type mechanism. So we're quite interested in looking into more detail at the role of both beta 1 and beta 3 in facilitating these trans and cis adhesions and how that might influence ephaptic conduction. So in summary, beta 3 modifies the nature of Nav 1.5 assembly. While it doesn't alter activation properties, it does shift steady state inactivation in order to increase channel availability and accelerate recovery from inactivation. We know that the transmembrane glutamic acid influences kinetics of domain 3 and therefore recovery from inactivation, and that the extracellular immunoglobulin-like domain is crucial for beta 3's properties on shifting this steady state inactivation, as well as some of the recovery from inactivation. And quite interestingly, losing just that single threonine residue in the extracellular domain is sufficient to abrogate the steady state inactivation effects, suggesting that that region is very important for alpha-beta and beta-beta interactions. And so the beta subunits, particularly beta 1 and beta 3, may be performing important cell adhesion functions which facilitate the role of Nav 1.5 in ephaptic conduction. And I'd like to say thank you to Dr. Tony Jackson and Professor Chris Huang, who have been incredibly supportive throughout all this work, various members of the lab who have contributed to different aspects, including Dr. Samir Hamaya, Joe Reese, Sahib Zaki, and Jennifer Irons. Our collaborators at Washington University, John and Wandy, who carried out the voltage clamp fluorimetry work, Greg at Papworth, who introduced us to this interest in threonine deletion, and the team at the Central Laser Facility in Harwell, without whom the storm resolution imaging would not have taken place. And to our funders, the British Heart Foundation and the Isaac Newton Trust. And thank you for listening. I'm happy to take any questions.